Hello, welcome to our MDICX on clinical diagnostic somatic reference samples. I'm Carolyn Hiller, Program Director for Clinical Diagnostics at the Medical Device Innovation Consortium. Our somatic reference sample project is entering a public comment period that will close on December 6th, 2018, and we welcome your input. I'd like to introduce our speakers for today's webinar. They will tell you more about the project and the opportunity for you to provide input. Our panelists today are Stacy Beck, PhD. She's Director of Personalized Medicine, the Acting Director at the FDA, and is on our MDIC Somatic Reference Samples Working Group. We also have J.D. Alvarez, MD, PhD, who's Head of Oncology Diagnostics for Janssen R&D. He's our Somatics Reference Sample Working Group Chair. And we also have Timothy McDaniel, who's the Senior Vice President of Emerging Opportunities, Professor of Integrated Cancer Genomics, Translational Genomics Research Institute, and City of Hope, also known as TGen. He's on our MDIC Somatic Reference Samples Working Group also. Please enter your questions in the comment box. We'll have a Q&A at the end of the session. Now I'd like to turn it over to Stacy Beck.
Good afternoon.
Stacey?
Yeah, good afternoon. Thank you all for joining us today. We are going to talk a bit about an effort that we're all participating in. But first, I'd like to introduce some of the efforts that FDA has undertaken to reconsider their approach to diagnostic testing when things are changing so rapidly. As Caroline mentioned, my name is Stacy Beck, and I'm the Acting Director of Personalized Medicine at the FDA. So traditional diagnostic testing generates one result per test. Think about running a glucose test or measuring someone's sodium. These might be done on the same analyzer, but they're looking at different analytes in a sample. A test report is usually generated and given to the physician, telling them what their patient's values were, along with some reference values for that test. As technology and knowledge surrounding genetics have advanced, as technology and knowledge surrounding genetics have advanced, this traditional model no longer holds. With next generation sequencing, one test can provide volumes of data, which often cannot be predefined or the disease identified until after the test is actually performed. This steps away from the one test, one result model and turns to a one test, thousands of results situation. While the agency is adept at reviewing the first model, we have to reconsider our approach when faced with all of this information in the second model. What we have realized is that much like you wouldn't take the prehistoric Flintstones car onto a modern superhighway, we realize we can't use the same old review models when we are operating in this new world. We are trying to regulate NGS technology-based tests in a way that enables innovation. Sorry, there we go. Okay. We are trying to regulate NGS technology-based tests in a way that enables innovation, requires a nimble regulatory approach, which is an approach that can keep pace with the scientific and technological advancements in this field. Ultimately, I'm sorry, this is really sensitive on the clicker. Okay. Ultimately, our vision is to accelerate the translation of precision medicine technologies into treatments that benefit patients. We are trying to provide an efficient path to market for these technologies while making sure that patients receive accurate and meaningful information. Caroline, do you want to take control of the slides and I can just tell you next slide?
Sure.
Because it seems like sometimes I'll click it and it's not going forward, and then I'll click it again and then it goes forward like 2 or 3 slides.
Of course. And are we on the slide you'd like to be right now?
This is the correct slide right now. Okay.
Okay.
Thank you. Okay. So how have we been trying to enable this nimble regulatory approach? We've really kind of approached this from 3 different prongs. The first part of the approach details technical standards for NGS-based tests. We envisioned that these standards would be developed with the scientific community and can be updated as science and technology advance. I will go into further detail on these technical standards in the next slide. The second part of the approach outlines a process where a public database of human genetic variants could apply for FDA recognition and can serve as a source of valid scientific evidence that test developers may be able to use to support the clinical validation of their test. And the third part, of which you'll hear more about from JD and Tim, is to promote the development and characterization of publicly available NGS reference sample sets that can be used to support validation of NGS platforms. And you can move to the next slide. So let's go ahead and dive into the guidance on design, development, and analytical validation of next-gen sequencing-based IVDs. This guidance was finalized in April and was specific to targeted or whole exome sequencing NGS-based tests intended to aid in the diagnosis of individuals with suspected germline disease or other germline conditions. The guidance makes a series of technical recommendations that can form the basis for either FDA-recognized standards or FDA special controls created in the future. The design section describes the test design approach, starting from the purpose for testing, steps to follow, what is needed, and what is available. You will note that the recommendations on how to design the test are very flexible. They can accommodate different test designs and different indications. The final guidance recommends that test developers predefine, justify, and report minimum acceptable overall and target threshold metrics. We do expect that specific numeric thresholds will be defined and specified in either upcoming consensus standards or special controls that would be developed by the FDA. We also include recommendations for what to present in test labeling. We believe that the recommendations in this guidance can be taken on by a standards development organization and that they can form the basis for consensus standards that can be developed by relevant stakeholders and experts. Standards developed by accredited consensus standard bodies are regularly recognized by the FDA. Of course, a big advantage of these standards is that they can be updated as technology and knowledge advances. Conformance with FDA-recognized consensus standards can be used to support a reasonable assurance of safety and effectiveness for many applicable aspects of these tests. We envision relying on compliance with FDA-recognized standards that would guide the development and validation of NGS-based tests and can replace some of the pre-market review to provide a reasonable assurance of test validity. The guidance also lays out a potential path where NGS-based tests intended to aid in the diagnosis of suspected germline disease could be considered as candidates for down classification to Class 2 devices. All novel tests are Class 3 by default, but FDA continues to believe that this may be possible in the future as we gain more experience with these devices and standards and special controls are developed that would help provide a reasonable assurance of safety and effectiveness of the test. You can move to the next slide. The second prong is to build a program to recognize genetic databases that are publicly accessible, transparent, and have an appropriate level of scientific rigor for use as sources of valid scientific evidence to support clinical validity of genetic tests. This is a voluntary recognition pathway, but we believe that it would be very beneficial to sponsors since it introduces regulatory simplicity into the process of genetic test innovation and availability for patients, and it reduces the regulatory burden on both industry and the agency. It will help change the paradigm of proprietary genetic databases, which can often suppress technological innovation by encouraging data sharing and publicly available databases of evidence supporting the clinical validity of genomic tests. You can move to the next slide. The third prong to FDA's approach to ensure a nimble regulatory environment for NGS tests is to help promote reference samples. We believe that if there are well-characterized reference samples, these can be used as truth sets to help compare platforms and provide evidence to show performance of a platform. These could be used to reduce the number of clinical samples needed to support regulatory review, particularly for variants, which are hard to find. Precision FDA is also a platform that we support that can be used to analyze, integrate, and compare data sets. We do believe that this is a community effort and not something that FDA should undertake alone. We have been involved in a number of these efforts in the past. Many of these are shown on the next slide. Caroline. So many of the different efforts that we've been involved in are shown on this slide. I'm going to speak a bit more about our interactions with NIST on genome model and MDIC on the somatic reference sample effort, but there are many of these that have been undertaken. We can move to the next slide. So CBRH has supported the Genome in a Bottle effort since 2012, both in a financial and in a professional sense. In this case, NIST is really developing genome reference materials, and they've actually already created 5 reference samples, and they're continuing to extend these efforts. You can move to the next slide. FDA has also joined with MDIC to develop cancer genomic somatic reference samples. MDIC and FDA have worked together on several projects, and this partnership has already been fruitful in many other areas. MDIC is really leading the effort on the somatic reference sample project, but it does include representatives from academia, pharma, CMS, NIST, FDA, CAP, ATCC, the device industry, and professional organizations like AMP, AACR, and others. We really wanted to make sure that there were all those different stakeholders were a part of this effort, because we think that to really be successful, we need the whole community to be involved. We believe that once these samples are developed, they can be used as an aid in efficient NGS test development and validation. They can streamline and possibly remove steps in the regulatory process for these tests, and they can provide transparency and compress development timelines for specific therapeutic developers. I'm going to hand this over to JD so he can talk more about the MDIC in this effort.
Hello, thank you, Stacy. So I'm JD Alvarez, and I am a pathologist by training, so I'm a longtime customer of diagnostics. And I currently lead the diagnostics group within Janssen Oncology, which is the pharmaceutical arm of Johnson & Johnson. And if I can advance the slides.
Okay, JD, do you want to try and see if you can control them from yours, or would you like me to just continue advancing them?
I guess, why don't you just go ahead, since that seems to be working.
Okay.
So next slide, please. Yes, so to level set everyone, for those who might not be familiar with the MDIC, the Medical Device Innovation Consortium, it's a nonprofit, and it's the first public-private partnership whose sole objective is to advance regulatory science. And it brings together a number of different stakeholders, including government agencies, nonprofit groups, academics or associations, and industry partners being diagnostic or pharma. So next slide, please. So what is regulatory science, since the MDIC is built to advance regulatory science? This is the science of developing new tools and standards that allow FDA products to be assessed for safety, efficacy, quality, and performance quicker and better. And to me, that's a very laudable goal, because when that happens, we are able to bring innovative products to patients quicker. We're able to increase the quality of diagnostic medicine and make sure that patients are getting the right test and the right drug. So next slide, please. So the MDIC has a number of different initiatives ongoing, which Carolyn could talk much better about than I could, but this is a list of them, and we're more focused on the clinical diagnostics and building regulatory science around that. So if we go to the next slide, please. So out of that was born the Somatic Reference Standards project. This is an overview of it, where we have, much like everything in MDIC, we've brought together a number of different stakeholders, government agencies, academic medical centers, payers, biorepositories, professional societies, and then finally, diagnostic and pharma industry. So the next slide, please. So the Cancer Genomic Reference Samples project, or SRS, has a very simple goal. which is to improve the efficiency and cost effectiveness of developing NGS-based tests and validating those tests by establishing needed reference samples. And the benefit of the outcome of this work will be to streamline test development and ideally obviate steps in the regulatory process so that diagnostic companies can bring novel tests to market sooner. Next slide, please. So the SRS is divided into two phases. The first phase is to publish a detailed description of the prioritized set of samples and variants that we want to characterize. So, and that is something that Tim is going to present on later. Once we've decided what are we going to do, what variants are we going to focus on, then we need to make a plan for actually how are we going to make them and how are we going to characterize them, which will be carried out in phase 2. And the MDIC will determine whether to continue to phase 2 on its own or to transfer to a third party. Then we are targeting to have phase 1 finished in 1 year. Phase 2 consists of actually making the reference samples. So we'll need to contract and oversee production and characterization of the high priority reference samples that we decided upon, and transfer these outputs to various commercial entities that would want to make these available as a product. Next slide, please. So this is the team. We have a core team consisting of myself as the chair, Giovanna Tezek from the FDA, CDRH, Julie Snyder from CEDAR, Stacy Beck, who just spoke, also from the CDRH, and Carolyn is the project director. And then you see we have representatives from a number of different groups, academic societies, pharma, government agencies, device manufacturers, academics, some suppliers, and various affiliates like Palmetto. So the next slide, please. So we kicked this off back in June. So it's been about 4 months at the FDA. And out of our kickoff, we formed 6 working groups. The Landscape Analysis Group, Variants Prioritization Group, again, which will be presented more by Tim, the Sample Definition Group, Business Plan Group, Sample Quality and Validation, and Data Integration. And I have these listed into the phases, not to say that they're only working during those phases, but that's where the bulk of the work will happen for these different groups. And we very recently had a progress meeting at the MDIC where we presented preliminary results of landscape analysis, sample definition, and the variants group. Started making some tentative plans for the other working groups. So next slide, please. So these are the questions that we needed to answer and are still in the process of answering. But you know, the landscape analysis group is focused on what's actually been done, what's ongoing, because we don't want to duplicate efforts. And there is quite a bit of work going on in terms of making various reference standards. We also need to decide what are the variants that are most important to develop, and that's the reason for this webinar, is to introduce our preliminary prioritization list for public comment. Once we've decided what we're going to make, then, or what we're going to focus on, then we have to decide what's it going to look like. What will those samples be? Will they be FFPE? cell lines? Will they be tissues? Those all have their pluses and minuses. So that's ongoing work by the sample characterization group. And more in phase 2 is work how are we going to validate those reference samples, and finally how are we going to make them available. We have decided to focus very specifically on solid tumors, and we are not focusing on circulating tumor DNA at this time. So next slide, please. So just quickly to go over the different working groups. So the Landscape Analysis Group is chaired by Barb Zenbauer at Emory, and they've done quite a bit of work developing a summary to identify efforts that are ongoing, developing and evaluating various reference standards. They've conducted a comprehensive review. They're planning to publish that sometime early in the new year. They did this by surveying NGS users to find out what's going on, but also to understand better what users really need. The next slide, please. The Sample Definition Group, chaired by Mary Ellen DeMars at ATCC, is to really define what are the reference standards going to actually look like, what physically. And this could be specific cell lines, it could be in silico, it could be a combination of both. And they're developing characterization criteria, acceptance criteria, quality control specifications around that. And then finally, they'll develop strategies to maintain a supply of the reference standards over time and ensure that they are stable. So they have made progress in developing minimal requirements for reference standards, encompassing a number of different aspects like consent and licensing. And they're performing an in-depth evaluation of current samples and attributes from commercial sources and various initiatives. The next slide, please. The Variant Prioritization Group is chaired by Tim McDaniel from TGen, who's going to speak after me, and Eric Peters at Genentech. And their mission is to define a list of clinically actionable alterations that would be represented in the ideal set of somatic reference samples. And Tim is going to talk about that today. The next slide, please. Data quality and validation, chaired by Jason Merker at UNC, is to determine a plan and how they'll experimentally verify the presence or absence of somatic variants within the reference materials that we made. They have a number of deliverables. They'll essentially make a plan in phase 1 and develop a budget so that this can actually be carried out in phase 2. The next slide, please. The Data Integration Working Group is chaired by Li Tai Fang at Roche. They will be taking data from the reference samples and determining what ground truth is for those samples. So they are currently developing an analysis proposal and a plan for how they will actually determine the ground truth. And again, most of that work will be done then in phase 2. Next slide, please. The business plan is chaired by myself. So we need to lay out a plan that will entice groups that would want to invest in the development and characterization of these samples and variants, essentially make a business case why this should be done. So we're in the process of gathering data, but the bulk of the work will be started in the new year. The next slide, please. This is our timeline. So you see that this year in 2018, The landscape analysis, variance prioritization, and sample definition groups have been working very diligently and have output to show for that. The data validation and integration, well, they've been will be ramping up primarily in the new year. Right now, they're planning and then getting ready for phase 2. Similar for the business plan, we are targeting to have that complete by June. The next slide, please. So with that, I will turn it over to Tim.
Thank you, JD. Right. Tim, you should be live now.
All right. Thank you, JD, and thank you, Carolyn. Yes, so let me come right out and say that my talk is an advertisement. My goal is to make you aware of the public review period for the SRS Variants list and to solicit your feedback before we finalize it. So today I'll be explaining how the team was organized, what principles guided the list's development, and then provide instructions for how you can access the list and send in your feedback during our public comment period. And Carolyn, next slide, please. So defining the variants has been the first order of business for the larger SRS team. After all, how do you define a set of reference samples without first defining what variants need to be there? Next slide. The Variants Working Group, like the wider SRS, has broad representation from a wide swath of stakeholder organizations. And I'm pleased to say that these members are really engaged. People have actually been attending the standing meetings and have been very active in producing, debating, and signing off on our work. Next slide. Our team's remit and approach are stated here. The team's task is to identify the list of variants or other targets, for example, genomic signatures, that would be covered in an ideal set of reference materials. In an effort to ensure that this list identifies the most useful targets to benefit the most patients today, we will draw from the targets from work of objective third parties without vested commercial interest in any particular target. Such sources should therefore represent current standard medical practice, originate from a public sector or independent nonprofit organizations, and represent the collective opinion of a broad swath of clinicians and or laboratory scientists. So using these principles, the sources we ultimately chose were OncoKB, Civic, and the Cancer Genome Interpreter. And to provide the effort focus, as JD mentioned, the initial list will focus on variants relevant to solid tumors. Next slide. In defining the variants, we recognized early on that we were up against two fundamentally different tasks, and so we broke the effort into two work streams. For hotspot mutations, the task is simply to define a list of clinically relevant variants that recur in cancer, including small variants, copy number variants, and rearrangements. This effort was led by Dan Klass at Roche. For tumor suppressor genes and genomic signatures, the task is different, as the number of potentially relevant mutations is so large that there would be no hope of representing them all in a reasonable number of reference samples. So the goal is to define a set of variants representing diverse mutational classes and genomic contexts, such that if you can demonstrate your proficiency in detecting these in a platform validation, then you will have demonstrated a general capability to detect whatever might come up in a clinical case. This effort is being led by Michael Morrissey at Merck. So as you can imagine, the second task is less straightforward than developing a simple list of variants, and so the first effort is further along. Therefore, the rest of my talk will concern the hotspot effort, and we will look forward to sharing more about the tumor suppressor gene and signatures effort in 2019. Next slide. With the hotspots, we're prioritizing our efforts to identify variants with levels 1 and 2 clinical evidence as defined in CDRH's tiering system for NGS. Level 1 represents those variants that are associated with approved companion diagnostics, and level 2 represents mutations with evidence of clinical significance, such as by inclusion in guidelines. The team is also generating a list of level 3 mutations, but this will not be delivered in the same time frame as the others and will not be part of the near-term public review period. Next slide. The process of generating our hotspot list has occurred over 4 months, with 2 face-to-face meetings, bi-weekly teleconferences, and much work over email from the entire team. Next slide. Our current status is that our level 1 and 2 variants list is released for public comment today, going through December 6th. The team will incorporate the received feedback into a revision, and the final list will be completed at a January 23rd face-to-face MDIC meeting at the FDA. And of course, at the same time, we will be posting the final results online. Next slide. So how can you participate in this process? You may access the list at the URL listed here, and then upon review, share your comments with us by the December 6th deadline through an email at the address listed here. The final list will be available for public review on January 23rd, and if you're scrambling right now to write these addresses down, please hold on, and in a minute, I'll get to an easier way. Next slide. Now, during the public comment period, we will ask the following 3 questions. Number 1, are there any variants missing from our list? And if so, why? Number 2, is there a variant on the list that should not be on the list? And again, if so, why? And finally, are any of the variants miscategorized? For example, a level 1 variant that we've called a level 2 variant. And again, if so, why? Next slide. So this is a snapshot of the first 14 lines of our variant list. And I draw your attention to the first column, which for every variant, there will be a variant unique identifier. And as you provide feedback, we ask that you reference any variant by using the variant unique identifier number. This will help avoid ambiguity. Next slide. Now, as I promised, those long addresses a couple slides back, if you're more the navigating type, you can find them here at mdic.org. On the landing page, click the Projects drop-down menu and select Clinical Diagnostics. Once on the Clinical Diagnostics page, select Somatic Reference Samples. And then once there, you'll have access to the variant list, instructions for comment, and other related documents and lists. Next slide, please. So that's the SRS Variants team, the process we followed, and the resulting variants list. The public comment period is now open, and we look forward to your review and comments. So I'd like to thank the entire SRS Variants team, Carolyn Hiller, and I'd like to thank you for your attention. Thank you all very much, and now I'll hand it back to Carolyn so we can start the Q&A session. Thank you very much.
Thank you, Tim. I'm going to unmute Stacy and JD. So all of our panelists are currently unmuted, and I do not see any questions that have been submitted from the participants. Tim and Stacy and JD, I guess I ask you if you have any closing thoughts that you'd like to share. Again, we invite everybody to provide the input that is spelled out on the web page that Tim had referenced. If you've missed them, if you Google somatic reference samples, I think you'll find the web page is the top hit for the somatic reference sample project. Stacy, Tim, JD, any closing comments?
I would say, yeah, I want to echo Tim. I want to thank everyone for their participation. I would ask that please tell your friends and family about this. You know, Tim's group has worked extremely hard on developing a list of variants that they think is comprehensive, but we need as much feedback as we can get so that we can ultimately decide what's the product we're going to make. So the more feedback we have, the better. So please put in your comments, and please ask everyone you know that might be have interest in this to also do the same.
Thanks, JD. If anybody has any other questions, feel free to reach out to me at MDIC. And thank you very much for your attention, and we look forward to your input.